In this video, we're going to take a look at several ways we can analyze DNA. One way that scientists can analyze DNA is by searching the National Center for Biotechnology information. Whenever scientists sequence an organism's DNA, they typically will deposit the DNA sequence in a repository here at the NCBI. There is a program that we can use called BLAST that allows us to search the NCBI in order to identify what DNA sequence comes from a particular organism. And this BLAST program is named for Basic Local Alignment Search Tool. So this is a screenshot of the BLAST screen where you simply put in your sequence and then you can blast it and it will search for the organism whose DNA you've just entered. So let's take a look at an example of how we can use BLAST. I have here a particular sequence of DNA that I don't know from which organism it comes from. So I'm going to copy the sequence and then go to my BLAST site and then click on Nucleotide BLAST. I then paste in my sequence, scroll down, and select BLAST. Now it's searching. And then when the results come up, we then scroll down and we take a look at what name appears. And in this case here, my organisms, the DNA that I have searched comes from Pseudomonas aeruginosa, a bacterium. I can also use the BLAST site if I wanted to align and compare two different sequences of DNA to see what portions of the sequence is identical or what portions of the sequences are different. So I will put one of my sequences as the query sequence and my other as the subject sequence. And BLAST will make that alignment for me, and you'll get something that looks like this, where it will indicate the identity, so how many of the nucleotides do in fact match up. If I have two identical sequences of DNA, then the identity should equal 100%. And then it will actually show me that alignment. So in this case here, if we look at the first nucleotide, you can see that both of them have T, so they match up. While over here, the query sequence has a C, while the subject sequence has a T. Therefore, they are not matching up, and there is no line connecting them. I have an example that I can show you of this. So here I have two different sequences of DNA, and I'm interested in aligning them to see if they are in fact the same sequence or what portions of these sequences are the same. So once again, I will highlight them, and I can include the name in it. It's not going to be read by the BLAST sequence as long as you have the caret symbol next to the name. So I'll copy this. We'll go to our BLAST site again nucleotide. And now this time I want to align two sequences, so I'm going to click the align button. Actually, I have to paste them in one at a time, so let me go back. We'll take sequence 1. Paste that in. And then sequence 2. And paste that in. And it doesn't matter which one you use as the query sequence or the subject sequence. And then again hit BLAST. And now we look. We see that the identity reads 79%, so that shows me that those two sequences of DNA were not identical. And as you can see here is the alignment where it shows you where nucleotides of DNA are the same. So as you can see, this stretch of nucleotides does match between the query and the subject, while here we have some insertions. Over here we can see that one has a G while the other has an A. Since we have this power of aligning sequences of DNA, or we could do this with RNA or with proteins, we can use these alignments to look at molecular evolution and to prepare evolutionary trees. So see, here we have an example of an alignment, and as you can see that all of our sequences have this TAA. So that shows there must be something common amongst these sequences of DNA. However, looking over here, you can see that these two sequences have T, while these three have C, and the rest have A. So that must show that there must have been some difference that has occurred within my sequence of DNA. And an evolutionary tree is pretty much a graph, if you will, of that alignment. And the branches in the tree indicate the different sequences that correspond to different species. So the more close together two species are in the tree shows that they are more closely related. So for example, if I look within the eukaryote domain, the diplomonads are more closely related to microsporidia as compared to, say, the plants, the fungi, or the animals, since they are much further away than microsporidia is to diplomonads. So there's a website that we can use to perform this alignment and to prepare these evolutionary trees. It is a site called Muscle, and I'm going to show you how that site works now. So here I have the DNA sequences to different species of lizards, and each lizard name is indicated here, followed by its DNA sequence. So I'm interested in preparing an evolutionary tree of all of these organisms based on the sequences of DNA that I have here. So I'm going to copy all of my sequences and then go to the Muscle website. I'm going to paste in all of my sequences, hit submit. And wait for the alignment to complete. Okay. So here's our alignment, and now I'm interested in seeing the phylogenetic or evolutionary tree of this alignment. So I'll simply go here, and when I click, it prepares an evolutionary tree for me. All of the links that I've shown in today's video will be listed in the description below the video.